We will learn about microsporogenesis. Microsporogenesis is the process of formation of microspore in microsporangia. As I told you, in the case of flowering plant, microspores means pollen grain. And these pollen grains are produced inside the anther of stamens. You also know stamen is the male reproductive part of the flower, and it has two main parts. Long stalk, that is known as filament, and upper box-like structure, it is known as the anther. And you also know flower is a modified stem, hence the part of flower such as calyx, corolla, endoecium, gynoecium means sepal, petal, anther and gynoecium or carpel. They are the modified leaves. In this way, this stamen is microsporophyll. Microsporophyll which contain microsporangia, and inside the microsporangia, microspores are formed. To understand process of microsporogenesis, we first observe the structure of anther. You know, in the case of flowering plants, anther is called bilobed or two-thecus or bilobed or dithecus structure. Now observe this transverse section of the anther. These are the two lobes, one lobe and another lobe, and these lobes are separated by a tissue that is known as connective. Each lobe has two sporangia. These are sporangia. It means one lobe has two sporangia, and inside these sporangia, spores are formed. Now we take the structure of microsporangium. As I told you, microsporangium appears circular and surrounded by four layers. The outermost layer is known as the epidermis. The cells of epidermis are protective in nature. It means it forms a protective layer. Next is endothecium. This second layer, endothecium, has a thick walled cells and help in dehiscence and is protective in nature. Third layer is known as middle layer. It also helps in dehiscence and also protective in nature. The fourth layer is tapetum. It contains the cells with dense cytoplasm and many nuclei, means multinucleated. It provides nourishment to the developing microspores or pollen grain. And in the center of the microsporangium is filled with closely arranged identical cells called sporogenous cells, and this group of cells or bunch of cells is known as sporogenous tissue. These sporogenous cells are further developed into microspore mother cells, and then by the process of meiosis, these cells form microspores. Now we will learn about the process of microsporogenesis, means how microspores are formed. Each cells of sporogenous tissue acts as potential pollen mother cells or microspore mother cell. This pollen mother cell or microspore mother cell undergoes meiotic division to form cluster of 4 cells called microspore tetrad. Means it is a microspore mother cell. It undergo meiosis, and after the meiosis, 4 haploid cells are formed. These 4 haploid cells are arranged in a group of 4 cells, hence it is known as microspore tetrad. In this way, inside the microsporangia, many microspore tetrads are formed, and after maturation, from these microspore tetrad, microspores are separated, and these microspores are developed into pollen grain.